bird lovers. Taking a look at bowers, at bird song, aesthetics and ornamentation and outside ornamentation. And we're going to continue by taking a look at the wondrous flight of birds. So I'm excited for us to investigate that topic. And there's a lot to learn. Biologically based flight is so foreign to the human experience. And I think that's one of the reasons why it fascinates us, and one of the reasons why we can gather here today and talk about it. I, for one, have been a bird fan since I was a little girl. I remember going out in the backyard and seeing what birds I could spot there, admiring the great variety of flight, the spread feathered flight of the crows, and the swooping flight of the evening swallows. It wasn't until college, though, when I had the good fortune of taking an inspiring ornithology course with Richard Prum, that I began to learn about the developmental and evolutionary mechanisms that make flight possible. And so I feel privileged to be here today giving this lecture on the theory of flight. So where to begin? Well, in order to fly, you probably need wings. So the question becomes, how exactly do wings grow? Well, the beginning of the understanding of wing growth was taking shape around 1948 with the revelatory experiments of John W. Saunders. And we have to remember that this is before the 1953 paper of Watson and Crick that defined the double helix structure of DNA. So this is before it was understood that DNA serves as the code to define the entire body. Different genes within that DNA sequence are expressed to form proteins, and you get different genes expressed in each cell. So you get a very different set of genes expressed in a skin cell compared with a nerve cell. And it's rather magical how each cell knows where it should appear. Your whole body grows in an organized fashion so that skin cells are appearing on the outside, and underneath that, muscle cells and nerve cells. Toes are growing in the right place, and arms are growing in the right place. So in 1948, Saunders was conducting experiments on chicks. And he found special cells in the side of the developing chick that seemed to define where a wing would grow outward. Now, this being before scientists were manipulating genes directly, Saunders was manipulating those cells. And he found that if you remove those cells, wing growth would be stunted. And if you move those cells to a new location, say the back of the developing chick, then you get a new wing growing in that new location. So these cells seem to be acting as organizers of wing growth. They later be called the AER, the apical ectodermal ridge. 20 years later, Saunders was still conducting experiments on chicks, and he and his colleague, M.T. Gosling, found other cells right at the base of the AER that seemed to define the orientation of digits on a limb. You move those cells to the top of the AER, and you get an upside-down wing, where the third digit appears where the first digit usually appears. In the 60-plus years since those original experiments by Saunders, there have been tons of experiments that have identified the genes within those special cells that are acting as organizers. Sonic hedgehog, SHH, turns out to be the gene that is crucial in defining limb orientation. So it's the gene that Saunders and Gosling were manipulating to get that upside-down wing. SHH acts as a signaling molecule. It binds to a receptor in the cell surface, triggering a cascade of protein interactions within the cell, ending with a protein binding to a regulatory section of DNA. That causes the DNA to unwind. RNA is then transcribed from that DNA and then converted into a chain of amino acids, which is folded in pleats and twists to get a protein. That protein can activate another protein, which activates other proteins, which activate other proteins, all as a result of that first SHH signal. And SHH, well, there's a project I've been working on, and SHH has been very important. And since we have so many bird lovers here, I really love to share it with you all. Yep. Do you see the feather? Well, it's been a sort of breakthrough in my work here, and I was able to activate the whole complicated growth of this feather by using SHH signaling. SHH is found in both birds and humans, and when I introduced avian genes into the novel hair follicle context, I was able to trigger this complicated growth by using endogenous SHH signaling. This is a complex structure. It's emerged from branching morphogenesis with a midline rachis and barbs branching off of the rachis and barbules branching off of the barbs and barbacells branching off of the barbules with little hooks that connect the adjacent barbules to create an aerodynamic feather plane. Well, it's almost aerodynamic. There's still a long way to go. There's a long way to go before achieving flight. And I'm not really sure what the next step should be. I'm not really sure how to grow a whole wing. I mean, how do you get the bones to be the right length and the same number of feathers on the left and right sides, and the bones and muscles and nerves all to interact? It's all very complicated. What would be wonderful is if I could find a gene that could turn on the wing network, just like SHH turns on the feather network, and I could just turn that one gene on, and it would send signals to the thousands of other genes involved in building a wing, telling bone genes and muscle genes and nerve genes the right place and time to be turned on. Maybe actually SHH could be that gene. I mean, it's found in the feather growth and in defining the left and right sides of the body and in defining limb orientation, and it's found early in development and defining the neural tube. I mean, there are all these known connections to the SHH gene. Look at all of them. You've got Hox genes, you've got growth factors, you've got this complex network of positive and negative feedback, and well, we know that SHH is very powerful. If you remove SHH, wing growth is stunted. The ulna and fibula fail to differentiate. You have that leg with just one digit moving around. But even if that is a pretty pitiful wing, it's still a wing, so SHH may not be our wing network gene after all. We have to take a look at this network again. WNT3A and beta-catenin. These could be promising genes. I think they could be. Okay, so they're connected to FGF10, fibroblast growth factor 10, so we have cell proliferation, that's very important, and it's very powerful. If you overexpress it, you get that whole new wing growing in a new location, and if you remove beta-catenin expression, then the wing and leg fail to grow. So it's very powerful, more powerful than SHH. I think it's, but if the wing and leg fail to grow, then we have another problem. It's very powerful, but it's not wing specific. So it can't be beta-catenin, can't be SHH. Okay, we have to reevaluate this network. TBX5. TBX5 also connected to this network, connected to the FGF10. TBX5 gives rise to a wing, and we have a separate gene for wing specificity. TBX4 gives rise to a leg. TBX4 connected to FGF10, and we get a wet. So this is very, very promising. TBX5, it's powerful. You remove it, the wing fails to grow, and if you overexpress it in a new location, then you get a whole new wing growing in a new location. So this is very promising. I think TBX5 has the wing specificity, and it has the power that we need to control wing growth. this is very, very promising indeed. Well, we still have the problem that these genes are acting during embryonic development, not in an adult. But, well, I seem to have gotten a bit carried away. You don't need to really take in this whole network, but I highly recommend the Saunders and Gosling articles. They're very fundamental texts. And, well, I have to go right now, but I'll see you in a little bit. Of an entire proto-wing. This is an incredible leap forward from the first feathered foray into network co-option. Here we have a fully formed wing induced by TBX5 and FGF10 activation of introduced avian genes. The genes were incorporated using the well-established replication-competent avian sarcoma, RCAS, transduction protocol. Multiple feathers have developed, connected to a co-opted skeletal system with functional muscle and nervous system connections. And, well, maybe I shouldn't be telling you this, but I had a dream. There was a salamander, an axolotl, and it was walking in a village at night, and its leg fell off, but it grew back again. It gave me an idea, a solution to the problem that avian limb regulatory genes act only during embryonic development. Humans shed and regrow their hair, birds molt and regrow their feathers, but the axolotl can regrow an entire limb even as an adult. When an axolotl limb is amputated, a blastema full of multipotent progenitor cells forms at the wound. Cells that are capable of turning into all the different cell types that make up a limb. You have nerve cells, muscle cells, skin cells, bone cells. It occurred to me that if I were to introduce axolotl genes as secondary transgenics, I could induce the formation of cells that could have a whole variety of cell destinies, cells that could regenerate and proliferate as a wing. I had success introducing avian genes, and I had success introducing axolotl genes. So I amputated my arm, and rather than the standard human wound healing mechanisms of blood clotting and migrating epidermal cells, an axolotl-like blastema formed with the potential to generate a wing. Now I am so close to achieving flight. Now it is simply a question of applying this procedure to both forelimb appendages and tweaking the cell proliferation pathways of it so we get a wing at a more feasible scale. And this should be straightforward compared to the multi-species transgenics. defined. Now it is simply a question of training muscles to achieve aerial locomotion. In 1879, Samuel Wendell Williston proposed that as dinosaurs began to evolve wings, they must have first used their wings in running, next in leaping and descending from heights, and finally in soaring. Lift generated by flapping wings relieves the legs of supporting much body weight, allowing for increased running and eventual takeoff. A running, leaping, flapping regimen should be able to develop flight in my case, too. There are some objections to this cursorial theory of flight. It's been predicted that any ancient bird would have had to run 3 times as fast as any modern bird in order to achieve liftoff. And we can imagine that the rate for hominid liftoff would well exceed this factor of 3. Some of the first birds seem to have been tree climbers and perchers, and flight could have well evolved with gravity as a helper. The first birds would have glided to the ground from their high treetop perches before mastering propelled flapping flight. One of the baffling things about flight origins is made clear from this vantage point. A half-flying state would hardly be useful when taking off from here. But how are wings supposed to evolve and develop without some intermediary stage that is not perfected for flight? What if you launch yourself prematurely? The running, leaping, flapping regimen may be ineffectual, but it's more reasonable than jumping from heights. The ontogenetic transitional wing hypothesis. The OTW hypothesis predicts that transitional stages and evolutionary forms correspond behaviorally and morphologically with transitional stages and ontogenetic forms, that is, in the development of flight within an individual bird. Studies conducted at the University of Montana Flight Laboratory analyzing the flight of the ground-dwelling chukarbird demonstrate that level flapping flight is preceded by a period of wing-assisted incline running. Day-old hatchlings are able to clamber up a 60-degree incline using their wings and legs. By day 8, they can ascend a 75-degree incline. By day 15, 85 degrees. And they can ascend a vertical surface with wings flapping by the time they are an adult. elements of the cursorial theory, for here I am running up a ramp with elements of the arboreal theory, for flight is made possible by the descent from the incline apex. I should be getting a bit of lift at this point. Here we go. Here, gravity helps to reorient the glen away from the dorsal cardinal position. The exalted to the dorsal longitudinal position. Well, attached to the exalted. Should be getting a bit of lift. Demonstrated in 20 avian species across the phylogenetic tree. Okay. Oh my God.